So I'm going to get started with introductions. We're very fortunate to have Dr. Jair Soares and Dr. Melissa Delbello today, both of whom are here for two main reasons. One, to talk about editorial perspectives on publishing in the field of bipolar disorder, and also to share their thoughts on neuroscience models that are relevant to bipolar disorder. So Dr. Soares will be presenting first. Dr. Jair Soares is professor and chair of the Department of Psychiatry and Behavioral Sciences at University of Texas at Houston. He's also the Pat Rutherford Chair. He has a number of leadership roles within his institution, both clinically and research-wise. From a mood disorders perspective, Dr. Soares chairs the UT Health Center of Excellence in Mood Disorders that focuses on research on the causes and treatments for mood disorders. He has an active team, highly productive, lots of mentorship focused on everything from genetics, biology, imaging, phenomenology. So welcome, Jair, and welcome everybody. And
well, thank you, guys. I'm honored to be here. So Ben, help me be sure that we are going to share this right. Just a sec. I'm going to look here. And is it showing correctly now?
it's showing perfect. Yep.
Oh, it is? Okay, cool. Great. Let me... Just gotta get my screens here correctly. Okay. Well, should we go ahead and get started then, Ben?
Over to you, Jair, for sure.
Excellent. Well, I'm honored to be able to talk to the Toronto group, and for the 20 minutes that I have, I want to give a brief summary of some of the research that has been ongoing over the past 3 decades, trying to find suitable, viable neuroimaging markers, for bipolar disorder. And some of that we've done, and Dr. Delbello and well, Dr. Goldstein has worked in this area as well, and many others. And I'll start by telling you that I can't help but feel that, I'm a little bit disappointed that after, 3 decades, it's still hard to pinpoint a marker there is, ready for to help us in the clinic, and I will discuss some of the reasons why I think that's so. I gotta okay. Did it advance? Are you guys seeing the next slide or not? Not really.
We're still on the first slide.
The first slide. So let me just how about now?
Good to go.
Great. Well, first of all, it depends on how we delineate the illness. As we know, we clinicians know there are different subtypes. And the boundary for what constitutes bipolar disorder is still hotly debated. Depending on how many days of hypomania you require, you will end up with a group of bipolar type 2. There is a lot larger. If you require just 2 days, it may be as much as 5% of the general population, which seems like a little too much. If you define more narrowly, requiring the 4 days that DSM-5 requires, you end up with something around 2% when you add BP1 and BP2, which, seems about right. But depending on where you delineate, where you put the boundaries, I mean, so that's issue number one, and different subtypes as well. I mean, as we know, the bipolars type 1, they have full-blown manic episodes, and the BP2s, just the hypomania. So some difference in the presentation and ongoing debate as to how these two things really relate. The other issue is that it's also very pleomorphic. The course of the illness varies a lot, and the clinical presentation varies a lot from one patient to the next. these are issues that could also account for much of the variability that we find, in the imaging studies that have been done over the past 3 decades or more now. Final issue also that is very relevant is all the comorbidities, medical comorbidities, psychiatric comorbidities, including alcohol substance abuse in more than 50% of these patients, which really, compound to the complexity of the clinical presentation and also, has to account for some of the variability for the brain imaging findings in this illness. We've been focusing on, these particular circuits here that I broadly call frontolimbic brain circuits. I mean, involved in regulation of our emotions. And we started out by trying to identify some regional changes with higher resolution anatomical MRIs and moving into trying to identify functional abnormalities at rest, as well as task related as the brain performs certain tasks, and more recently also a big focus on connectivity amongst these regions with DTI, diffusion tensor imaging, and quite a bit of literature showing abnormalities in these specific regions and circuits that interconnect them, as well as the function of these areas as, for example, people engaging to recognizing emotions or performing like a mental task that taps onto working memory as well. We've been part of, this large international collaboration called Enigma. This is led by Paul Thompson at UCLA, or now USC. And this is a summary results from a very large sample. And what seems to more consistently be found is a shrinkage in the amygdala, shrinkage in the hippocampus, shrinkage in the thalamus, and enlargement of the ventricles. So those seem to be the most consistently replicated anatomical findings, corroborated here in this large collaboration. Trying to do cortical mapping, essentially looking at differences for the gray matter content across different cortical regions. The letter A here is contrasting bipolar patients and healthy controls, and anything that is not gray is really significantly different. In this case, yellow, light green, and orange, those are areas of thinner cortex in average in the bipolar patients compared to an average group of healthy controls. Letter B, which is a whole different profile, those are bipolar patients on lithium compared to healthy controls. As you've seen, no areas of reduced cortical thickness, and some actual areas of increased cortical thickness, for reasons that have been debated over time, perhaps related to neuroprotective effects of lithium. Letter C here is contrasting bipolar patients on other mood stabilizers, in this case here, anticonvulsants, to healthy controls. And the pattern is more similar to what we found in the overall group of bipolar patients, compared to healthy controls, which is thinning of several, cortical thinning widespread across different cortical regions overall, and with some suggestion that it may even be more so than the overall group. And again, if you discount the ones on lithium, you may end up with effects that seem more pronounced. So here, same collaboration, just trying to look for specific profiles that would be more typical of bipolar patients. And for example, this is, same type of analysis done with schizophrenic patients. We will see lots of cortical thinning across, several, pretty much all brain regions. Major depressive disorder seems to be more restricted to certain areas. I mean, obviously, if you'd study very chronic patients with multiple episodes, you'd see a more widespread pattern of cortical thinning here. ADHD, kind of different, just like very light cortical thinning compared to healthy controls. OCD, whole different story. And this is autism spectrum disorders. So one would look at this and say, well, can't we come up with a reliable pattern that perhaps we could train a computer to recognize and therefore help us diagnose patients with bipolar disorder or confirm the diagnosis, upon suspicion based on an MRI. And that's what we and others, have tried to do. So before I go there, I want to show you also, this is from the same collaborative group, a different analysis and publication, but showing here that there is a relationship between the length of lithium treatment, well, lithium treatment for sure, but the length of illness. The patients who had been ill for longer periods of time with more episodes are the ones that have the more pronounced reduction in cortical thickness, and those tend to be the same ones where more refined neurocognitive testing will show more clearly neurocognitive abnormalities across, various domains of cognition. So this is sort of like broad-based, subtle in most patients, but broad-based. So we've also did a study here. Bo actually is in Canada, in Calgary. He was a former postdoc in our lab. And we had a group of unipolar patients, bipolar patients, schizophrenic patients, and healthy controls. This is not a longitudinal study, but we had sizable numbers, of patients across this diagnosis, and we did some cross-sectional analysis and plotted those as a function of age. Looking at differences amongst these groups across different across different ages. And the measure here is what it is called the Jairification Index. It's a more specific, it's a quantitative way to look for some like global changes in the brain, global atrophy, for example. It's a measure of the folding of the brain. So kind of like a global measure of quote unquote brain health. And what we see here that I want to show to you, the schizophrenics are in blue and bipolars are in green. Reds are UPs and blacks are healthy controls. So early in life, not much difference, but beyond the 4th decade of life, the patients with schizophrenia, as far as like this global measure of like brain folding, sulci folding, the schizophrenic patients seem to look more like the bipolars, and the unipolar patients are more in line with what you see for the healthy controls, suggesting that perhaps over time, there may be a gradual process here taking place in the brains of these patients. Then here I want to illustrate this notion of debated over the years that there could be some progression in the brain changes that one will identify, as well as the neurocognitive changes, that will reflect changes in the brain. And as a function of multiple episodes in higher stress load and the toll that this illness takes on the body and on the brain, and these folks, gradually having, in average, detectable neurocognitive abnormalities and brain changes, detectable brain changes, including anatomical changes like we discussed before. Key factors that seem to be involved in this neuroprogression. One is a history of trauma that seems to, significant trauma in the childhood is a risk factor. And the folks who had it compared to the ones who don't seem to have more pronounced neuroimaging changes as well as cognitive changes. When you measure those, in their adult age and look into the effect of the previous trauma exposure on those measures. Several studies have shown that. Number of episodes, as we discussed, seems to cause these changes to progress gradually. And substance abuse, that's a big factor that we know, makes the illness more complex and makes the brain changes more pronounced and makes also the recovery a lot more complicated. Well, what could cause this neuroprogression? What's been discussed over the years, neuroinflammation, oxidative stress, abnormalities in neurotrophin production, and others have also proposed the hypothesis of some level of mitochondrial dysfunction. All these are current and being actively investigated. We also know that these patients with bipolar disorder, they die younger. It's by 15 to 20 years. It really takes a major toll in their expected lifespan. And so there is evidence for some accelerated aging. And this can actually be detected across different biological measurements, not only with brain measurements, but immune measurements in these folks, and epigenetic measurements and measures of oxidative stress. The telomere length shortened in average compared to individuals who don't have an illness like this, essentially showing the toll that it takes on the brain and corroborating this notion that these folks, for individuals of a similar chronological age, their body, seems to be aging a little bit faster. If you look at the genetic inheritance, that gives me some consolation. It's no easier than this picture that the neuroimaging studies has revealed, in the sense that, many specific genes seem to have an effect, and mostly modest effects, modulated by epigenetic factors like genes getting activated or deactivated, and not a whole lot of replication from one sample to the next, despite the fact that now these studies, have involved, now it's like 20,000, 30,000, There was the hope that as the samples would grow, perhaps our accuracy would improve, but it's a very complex puzzle with many different genes potentially conferring a risk, as well as genes that may be conferring some protection as well. Well, I want to briefly show you some illustrations here of areas where in neuroimaging, if not at a point that has helped us diagnose, perhaps helping us predict the course of treatment, or target, treatments more specifically. Again, from work by our group here, Benson Wanji led this one. We tried to train a machine learning algorithm to see if it would get to a point of predicting with high accuracy based on MRIs, the patients that were bipolar. What we saw, we had to split the sample between kind of like early course, intermediate course, and late course. Late course, multiple episodes. This was based on the number of episodes they had. Early on, the model does poorly to separate those MRIs from the healthy controls. As does better on BP1s than BP2s, but pretty much as these folks have had more episodes and therefore more pronounced brain changes, that's where the model performs a little better, but still accuracy like in the 70s, and leaving us with the question as to how meaningful that is and can that be helpful clinically. And this is what our Enigma collaboration tried to answer again here. Very sizable sample size across 13 sites. Same question, trying to train a machine learning algorithm to recognize the bipolar MRIs and separate them from the healthy controls. We could not reach an 80% type of accuracy. It was, in the 70s. And again, leaving us with the question as to, does it justify the expense? If you do an MRI, the expense involved to separate those from healthies, which is not actually, clinically, most often we try to differentiate those from unipolars or patients with schizoaffective disorders and other kinds of psychiatric conditions. So the relevance still needs to be established. Well, here in our group, we are now doing a study. This is funded by the Milken Institute, part of the Barzuki Fund. And the idea here, we had some DTI, interesting DTI data from our patients with bipolar disorder, and we had measurements of anhedonia in these patients, a dimension of depression, which is like the inability to experience pleasure. And we focusing on the uncinate fasciculus, which is, a part of the brain important in reward circuits. We had this interesting findings that the ones with anhedonia had lower FA measures, showing either some changes in the tracts in that area or perhaps some abnormalities in myelination compared to the ones also depressed but without anhedonia. And so the new study is to utilize a technique called TDCS, transcranial direct current stimulation, and use the tractography measurements as guidance. And we want to do imaging before and after a course of TDCS. And this is still an experimental modality. Some studies have shown the results in depression, others have not. We want to see if anhedonia improves, and if it improves in the ones where the DTI measurements in the uncinate fasciculus, if those will be the determinants of improvement using this targeted finding in the brain that we have. So exciting work ongoing. I mean, we've done a few patients. We need several more, to have some numbers to show to you there. Final thing I want to show you. I mean, we also have had a deep brain stimulation study ongoing. In this case here is actually for very severe unipolar patients. We wanted to start with the unipolar patients. The target we picked is a medial forebrain bundle. This is trying to replicate Tom Schlepfer's work from Germany, where they had shown some interesting results from that, the modulation of this particular tract that interconnects all these important regions here involved in modulation of emotions and reward processing in the brain. This is just an illustration of where the electrode is placed bilaterally in these patients. And the short story here, and this is preliminary data that we published last year, and essentially showing that about 60% of these patients, for the ones that we had followed for at least 2 years after the implantation of the TBS device, focusing on the medial forebrain bundle, 60% response, which is pretty good, considering that these folks had failed ECT and pretty much all sorts of medications and psychotherapies available. We are now beginning to experiment with this ultrasound neuromodulation technique that allows for stimulation of deeper brain regions. In this case, we try to stimulate the amygdala, very early work. One of our postdocs here, Andrea Boscuri, is taking this work forward. He just got a NASA grant to do that. I just want to finish by saying that neuroimaging, so far has been elusive as far as helping us diagnose bipolar disorder. It may inform specific targets for treatment. We have the hope that it will help us towards this goal of precision medicine. I know that Dr. Delbello, has done some treatment trials looking for specific targets. She will have some good stuff to tell us about that. So Ben, since I'm now at 25 minutes, do you want me to? I have 2 slides about Jair. Should I save them for the end, or do you want me to go and cover the 2 slides of Jair right now? What should we do?
Yeah, why don't you go for it? I see Melissa nodding, so why don't you go for it?
Okay. Well, so I've been now co-editor-in-chief for Journal of Affective Disorders since 2015. And more recently, the past 2 years, we have a spin-off called Journal of Affective Disorders Reports. And Paulo Brambilla from Italy and I are co-editors for both. And JED has grown quite a bit. I mean, we took it at 1,500 submissions per year. We closed last year at 6,700, so more than quadrupled. And the good thing is that we have a lot to choose from. The difficult part is almost dizzying as to how many manuscripts we have to screen through. That has led us to have to reject more than 80% of what comes in. Paulo and I, with the past year, we added 4 new associate editors, and they are very helpful. Otherwise, we couldn't function. Well, what do we look for? Things that are fatal flaws and low-hanging fruit. Something that is sloppy preparation. I mean, you look at the abstract and there are typos and it's not in the right format that JED requires. That is not, if you have 6,700 to search through, divide by 2 editors-in-chief and divide by the number of days in the year, that's how many you will see per day. So if it looks poor at the abstract, that's where we are going to end, So that's an important thing to be careful with. Well, then language issues, and people debate that. I mean, bottom line, we have no time to rewrite the manuscript for someone. And even if the data is interesting and you read through that and it's very poorly written, that might lead to a fast rejection there. The times when we had 4 times, less manuscripts through, we might, have been able to be kinder and just go back to folks and say, hey, engage some native English speaker to help you or some translation services. But that, it needs to be written in a way that people, can read without being painful. And if it is not, unfortunately, that one will be, in all likelihood, rejected. We talked about the need to attend to the guidelines. Otherwise, it looks like you just very quickly, you just reformatted everything on EndNote and send it to us. If you did not take the time to see what type of abstract and what are the specific headers, that does not, bode well for, you're taking the journal seriously. Well, now, in this new phase where the impact factor has increased quite a bit, I mean, it's close to 7 now, and that's why we have seen this exponential growth in the number of submissions. Small case series, case studies, there are some journals that publish case studies, so we just recommend that you go there. Uncontrolled studies, don't tend to fare well. Or if the topic is very narrow and not likely to appeal to our audience, and again, it's affective disorder. So if you send something to me on schizophrenia, unless there is a spin around the mood, probably, we are going to feel that, well, this may belong better in a schizophrenia journal. I mean, it's not what we mean by affective disorders when we talk about affective disorders. Or if you were doing like a translation of a scale that has existed for a while to like another language, we feel, well, again, this is not new and it's very regional. Maybe you got to publish this in a regional journal in the language, that this scale has been translated, or, probably not a good thing for the JED, not of great interest for the JED audience there. Well, then lack of novelty. That's a very common one. We got these papers, very large samples, but it's stuff that we've known for years. So there's really no novelty. Those, again, because the sample is large, and if they've been done well, they may fit in the spin-off, JED report, which is growing very fast, by the way. But if it is like a replication, third, fourth, fifth or sixth replication with a smaller sample, perhaps needs to be published, but not at JED, perhaps at JED reports or somewhere else. Negative results with underpowered samples. That's another example there. Well, but so it's really, taking good care with that initial, it's like the abstract, the introduction. If you don't cause an impression there, the editor may not really go any farther, and you may end up with your manuscript quickly rejected. And that's what I had. Melissa, apologies. Well, you have the next half an hour. Thanks.
Thanks very much, Jair. I think I'll encourage, because we're at the halfway mark, as you said, thanks for leaving us the full half. I'll encourage folks, if they have questions about your neuroscience findings or about editorial perspectives, they can put them in the chat, and maybe you could address some of those, or if not, we'll connect with Dr. Soares and get answers for them. But with that, we'll switch to Dr. Melissa Delbello. I just wanted to remind folks also that this session is being recorded, will eventually be posted, and we have CME credits available for those who are in attendance. So Dr. Delbello is a professor of psychiatry and pediatrics and the Stanley and Mickey Kaplan Chair of Psychiatry and Behavioral Neurosciences at University of Cincinnati College of Medicine. She got her medical degree at University of Rochester and did her psychiatric residencies also at Cincinnati, as well as at the Payne Whitney Psychiatric Clinic at New York Hospital Cornell. She obtained her master's degree in epidemiology and biostats and has co-authored and authored over 350 articles, served as principal and co-investigator on multiple grants from NIH and BCORI, foundations, etc. Melissa is a leading scholar and thinker in the youth bipolar area, like Jair, a neuroimager and also a journal editor. So Melissa, thanks for joining and pairing with Jair.
Thank you. Hopefully, can you hear me and see my slides?
We can hear and see.
Awesome. Wow. Okay. So, fortunately, a lot of what I'm going to say, Jair wonderfully covered in his comprehensive talk. So I'm going to try and hopefully hearing it twice, it'll stick. And so I'm a firm believer in that. So, but my focus is going to be, as Dr. Soares predicted, a little bit more on treatment and biomarkers in treatment, as well as more focused on pediatric bipolar, because I am a child psychiatrist. So hopefully, again, okay. So why are biomarkers helpful? Just to kind of put it in context, in pediatric bipolar in particular, there is still a significant lag time to accurate diagnosis. I think the pendulum has swung that we are underdiagnosing to some extent patients with bipolar disorder who actually have bipolar disorder. And I've seen this on the inpatient unit where, you know, it's sort of in your face that a patient might have bipolar disorder and people don't diagnose it. And so there's still a big lag time. And unfortunately, a lot of times patients undergo several pharmacologic intervention trials prior to identifying the right medication for the right patient. And how can we do that better? Because not identifying the right medication quickly oftentimes will lead to pile-on of medication, poor tolerability. Patients then get frustrated, they get poorly adherent, and then that leads to poor outcome. So it's in everyone's best interest to rapidly identify the best treatment for a specific patient. So that's why biomarkers are important. And then also, there are no clinical predictors of treatment response. We know, for example, that lithium may be the best agent in terms of, you know, suicidality, but we don't know what clinical predictors there are of treatment response. There really aren't any consistent predictors. And so we really need to optimize efficacy and tolerability of pharmacologic interventions and create a more personalized, streamlined treatment approach. So that's why I think biomarkers are important. So what Dr. Soares nicely covered comprehensively, some of the most consistent biomarkers, particularly for pediatric bipolar disorder. We know that there are certain gray matter abnormalities, but it seems that specific gray matter abnormalities are not very specific, and global gray matter abnormalities are not specific for pediatric bipolar disorder. They're present in several other disorders, in ADHD and major depressive disorder, at least in children. And so global gray matter abnormalities and even in specific regions may not be a useful biomarker that's specific to bipolar disorder. And we also know from several studies that we've done and you guys have done and Dr. Soares has done that structural abnormalities are not present prior to illness onset. illness onset. They're barely present. Some may be present, but they seem to be associated with this neuroprogression that Dr. Soares was talking about. That markers of illness progression, yes, but not of illness onset. So they don't actually help us diagnose patients, and they don't seem to predict, generally speaking, outcome, although we'll talk a little bit about that. And then there are more and more studies looking at graph theory and networks and connectivity, and they seem to very consistently identify that prior to illness onset, that there seems to be in those who are most at risk, prefrontal changes in connections to other limbic regions, particularly prefrontal amygdala connectivity. And unfortunately, as Dr. Soares mentioned, a lot of these studies are confounded by differences in mood states and pharmacologic interventions. I think it's really important to note that there is a whole field of at risk also. So how can we use these biomarkers to identify who's going to get the illness? And the advantage, I think, in child psychiatry is that we have that, that ability prior to illness onset to look at at-risk individuals. So where biomarkers can be useful, just to kind of put it in a more broad context, is, as Dr. Soares discussed, looking at diagnosis. So can we use a biomarker to identify who has bipolar disorder? Can we use a biomarker to identify who is at risk? Out of, typically it's done from familial risk. So you have a parent or a first-degree relative with bipolar disorder. Those are the people that are most at risk for developing bipolar disorder. So can we use neuroimaging as a biomarker to figure out who's going to go on to develop bipolar disorder? And sometimes, though, where I think it's most useful, and I'm glad that Dr. Soares has come to the same conclusion, is that looking at treatment prediction and who's going to either respond to a medication, or lately we've been using imaging as, can we develop models to predict who's going to actually have an adverse effect to a medication. So for example, we've done some studies recently in kids who have parents with bipolar disorder who have what we might refer to as prodromal ADHD or prodromal early major depressive disorder or anxiety disorders. And then we treat them. So we incorporate treatment. So for example, in those who are depressed or anxious, the most likely treatment for those individuals are antidepressants. And so can we predict who's gonna get what we call antidepressant-induced hyperarousal? And so that leads to either exacerbation or acceleration of illness course and makes them more likely to develop bipolar disorder. And so it would be useful if we could use imaging as a biomarker to predict adverse outcomes. So who would we avoid who has a parent with bipolar, and whom would we avoid using an antidepressant, and be more likely to intervene with something like family-focused therapy, which may be just as effective as an antidepressant, but avoids the adverse outcome of hyperarousal and does not accelerate the onset of bipolar disorder. And then more recently, using machine learning approaches, as Dr. Soares discussed, to combine factors and lead to successful prediction of treatment outcome. And we've done some of that research. In the interest of time, this is just a summary of treatments that have been studied, and there have been some biomarkers identified in terms of kids with bipolar disorder, as well as those who are at risk for bipolar disorder. family-focused therapy, like I mentioned, mindfulness-based cognitive therapy. There's been some studies looking at that as well. And then the medications that I listed. There have only been a couple of comparative studies. So I'll discuss a specific example with our lithium quetiapine comparative studies in first episode, Adolescents with Bipolar Disorder. And then some of the studies that Mani Popolari has done looking at Delbello versus risperidone. And so the most useful studies are those that have comparison between two treatments or between a treatment and placebo. Although placebo-controlled studies are more difficult to do these days because we do have efficacious treatments for mania and depression. So, and then we talked a little about the stimulants and antidepressants. This was a very nice comprehensive summary review article that I think Dr. Soares was the senior author on, that summarized the neurofunctional effects of pharmacologic interventions in pediatric bipolar disorder. And I think this is really useful in that you can actually see where there might be useful markers to predict who's going to respond to what treatments. Now, there's differences between looking at groups of individuals who respond and don't respond versus using it beforehand to go on to predict. I'm gonna enhance this person's likelihood of responding to a medication by randomizing them to this medication because this is their baseline abnormality. And that is the next key step that we have to do to really solidify some of these targets of brain regions that have been identified that as potential biomarkers or targets for treatment response. We've done a study that I just wanted to use as an example of comparing lithium and quetiapine and looking at whether we can identify morphometric predictors of treatment response. And we used a machine learning approach to do this. And what was most exciting for me about this study is that we did identify very specific and different biomarkers of treatment response to about 80% accuracy. So it's not 100%, but 80% accuracy. And then what we did is we looked at whether those markers would be useful to identify response to the other treatment. And what I thought was most interesting is that they weren't. They were specific to quetiapine treatment versus specific to lithium treatment. And I'm not going to treatment response. I'm not going to get into the specifics of what these markers were, but they were very different in terms of type of structural abnormality. So if you're more interested, please read the paper, because to me this was one of the most important studies that I think I've participated in, because we can identify the specificity of a marker by throwing it into the model and looking at whether it predicts other treatment response. So that is just one example, but I think another example of how we can identify better treatment biomarkers is to look at not all bipolar disorder is the same, as Dr. Soares alluded to. There's manic episodes, depressive episodes, but there's also symptoms within bipolar disorder. And this is where sort of the RDoC approach may have some value. But looking at a patient's specific approach to their characteristics and their symptoms, not all patients with bipolar disorder have the same constellation of symptoms. A lot of patients have several of these characteristics, but not all do. And we have become much better at identifying which brain regions are involved in which characteristics. And so by using this approach, can we more specifically identify treatment targets by using the approach on the other slide, looking at these are the treatments that target these specific brain regions. This is the clinical constellation of symptoms that the patients have. Maybe we want to use a medication that would be better or more effective at targeting, for example, the middle prefrontal gyrus. We do know that that is really affects social cognition. Well, very consistently, something like mindfulness-based cognitive therapy is useful for modifying insula and medial frontal gyrus. And so maybe that would be an optimal treatment for a patient who presents with social cognitive deficits. And so this is just kind of linking it by using brain regions. So basically, I think that is sort of what this is referring to. So if we can identify the presenting symptoms, identify the potential underlying brain regions, and then use that to identify what treatments target what brain regions, that may be one approach that is novel to identify biomarkers, but then also can improve treatment outcome. So I do think this is a strategy we need to take in future studies looking at biomarkers to identify more predictive and specific models that will predict outcome. I also think we need more comparison studies. So again, like I mentioned, comparing two treatments, comparing treatment to placebo when possible. Looking at specific mood states is very important. Using multimodal imaging to predict outcome. In our hands, that's not actually helped prediction models. So by combining something like DTI with resting state connectivity, can you improve the models of prediction? Generally speaking, we don't, but others have done this, and this has been done very successfully in schizophrenia. Maybe bipolar disorder is different, and maybe there's a reason it's a more dynamic illness to some degree, and so maybe that is why we don't have as much success. And then ultimately, the next step that we really need to do is to use these biomarkers to then randomize treatments, randomize patients to specific treatments that we believe, based on their neurobiological characteristics, that they would respond better to specific treatments. So that's how we're going to validate these biomarkers for treatment, is to then do those studies that can we improve response rates by using these biomarkers and having a priori hypotheses of the targets for specific treatments. So I'm going to switch gears for a couple of minutes and hopefully leave a couple of minutes for questions. But I am the action editor for one of the deputy editors for JCAP, the Orange Journal, as other people might know it. Also for bipolar disorders, the pediatric field editor for that area. And also serve on the Silver Journal, the Journal of Child and Adolescent Psychopharm, as a neuroimaging editor. So I've had some experience, and I have to say, for those interested, the best experience and the most kind of in-depth experience I've had is with JCAP. It really, it's a great group of people. So if people are child-related and doing work and interested in taking a more active role, I know Ben's involved. It really is a great learning experience. I learned so much from my co-editors. But these are some of the suggestions, and interestingly, Dr. Soares covered most of these, so I don't want to be repetitive, but some of the specific, I know that has been a focus in child psychiatry, has been racial and ethnic diversity in the population that you're in the sample that you're studying, and in the population of the general population that the illness affects. And so if there is a disparity, that is being commented upon. Particularly, there's a focus on that in JCAP. And so if you are not a diverse sample, or if you live in the Midwest like I do, where we don't have a ton of diversity, we're now being asked to address that in our samples. So I think it's an interesting approach, but it's something that is more and more leading to, particularly with genetics, because some of the biobanks are not diverse. It's leading to more and more manuscript rejection. I think in the prior years, some of these manuscripts might have been accepted. So that's one specific point. And then also following regulatory standards, and I know in Journal of Affective Disorders, this is something as well. So clinicaltrials.gov is very important to register your trial if you're doing a treatment trial beforehand. And if your manuscript doesn't consistently have the same primary outcome measure as you're reporting in clinicaltrials.gov, that will cause automatic rejection up front. And people do check this. And so that is really important as well. And using AI to write your manuscript is becoming an increasing issue, and that is checked. There is some software that is used to make sure that that is not done. I don't know the efficiency of that software, but that is also something that is becoming more and more scrutinized. So with that, I will end and maybe give a few minutes for people to ask questions.
Thanks so much, Melissa and Jair, for your convergent perspectives on editorial boards and manuscript submissions. And also, it's just great to have the two parallel perspectives on neuroimaging. I think I have time for a couple of questions. I'll ask one, which is, our field has moved from small samples and uncertainty about the reliability of our imaging findings to larger samples consortia. Now we're quite certain about our genetics findings that they're reliable. We're quite certain about our imaging findings that they're reliable. But the impact and the magnitude of effect of those findings is small. And so I wanted to ask you to think about how could we better balance the search for, reproducible, predictable, reliable biomarkers with something that actually makes a difference. Because I think right now what it looks like is small effect sizes, very replicable, doesn't change lives for our patients.
Yeah, I mean, I think I'll be happy to address that first. I think some of it is, what we're calling biomarkers are really like predictors. And now we have to really validate them and establish that they are biomarkers in real populations of that. So there's a difference, obviously, between a predictor and a biomarker. And I think we've been calling a lot these predictors biomarkers, but they're actually just giving us some preliminary evidence. So I think then now we have to take it back and have clinical utility. Like I was saying, can we randomize patients based on their imaging findings and get better efficacy so that it can have some effect on our patients? That would be my answer.
So Ben, I think it is an issue of, well, first, the fact that the changes found in the brains of our patients are mostly subtle, unless you're talking about very chronic people who have had multiple episodes, and the sensitive of the tools as well. If you look at the PET literature, positron emission tomography, like, for example, the attempts to image different inflammatory mechanisms. And the tracers aren't that great. And then when you apply that, you end up often with findings that are inconclusive, right, where there's not a whole lot of replication. I think anatomically it really has to do with the fact that, compared to the disorders or the diseases that the neurologists study and treat, ours, they come with like more subtle changes, mostly functional changes, and very non-specific types of changes. On the genetic side, a multitude of genes that seem to confer vulnerability or perhaps give protection, some a very complex puzzle, and leading, to this commonality between our field of neuroimaging and others, where unfortunately there isn't much, that stands replication and that we can take to clinic. I do agree with Dr. Delbello that prediction of treatment might be a more profitable area there for us. We'll see.
Thank you, Jair. If anyone has a question, feel free to unmute yourself or type your question in the chat. Do you guys have any questions for each other?
Well, I do have for Dr. Delbello, I understand the question, the issue about the diversity of the samples. And I guess, obviously, it has to, I guess, at the end of the day, what is scientifically justifiable? Like for the geneticists, for example, there is an argument to be made that you get more power if you get less diverse and do a different study with a different patient population entirely. So I don't think we want to go overboard, and reject good manuscripts because they were done in Pittsburgh or Cincinnati, where the population is like 90% Caucasian. I don't think it is fair to, lessen the science or publish the results at a lower ranked journal for that reason.
Yeah, I would agree. Although if you're identifying certain markers in a not diverse genetics biobank, for example, but the illness affects, a disproportionate other race, then or equal in races, then how representative are those biomarkers? And so I think, as long as there's some like addressing it or discussion of it, I think that is important, because people then would say, okay, well, these are great biomarkers for bipolar disorder, but they may only be that way in a not diverse affected population.
Thanks, Melissa, and for that thoughtful answer about the topic. I think to be continued on it. We're close to 1 o'clock here, or at 1 o'clock. Just wanted to thank you both for presenting, for showing up on time, for being so collaborative. And also wanted to mention that February 15th, we'll hear from Josh Rosenblatt and Nishrat Hussain on ketamine and psilocybin. So please join us for that. Thanks, everybody, and have a great rest of your week.